deliver a talk on respiratory allergies in childhood. Sir, you're live now.
Let me Society and Dr. Osman Yusuf for the very kind invitation to come through the internet and talk to you. What I'd like to speak about is the role that respiratory viruses play in the development of asthma in children, and the question whether epithelial cell responses to these viral infections are critical. So in this talk I'd like to start by showing you some epidemiological data that link viral bronchiolitis in early childhood to asthma development. I will comment on some intervention studies, and then I'd like to really think about mechanisms and focus on mechanisms through respiratory epithelial cells. Let me start with what I would regard as a classical study now. This is a study by Niels Sigurds from Sweden, who compared children who were hospitalized with bronchiolitis to children who in the same season didn't have any bronchiolitis. And she followed these children up for 18 years and asked whether they had a diagnosis of asthma. This is the graph on the left-hand side, where you can see that up to the age of 18, the rates of children with an asthma diagnosis increase to just under 40%. On the right-hand side of that graph, you see the children who did not have bronchiolitis, who have substantially lower rates of asthma. On the right-hand side of the slide, you see another graph that also shows that the bronchiolitis children had higher rates of allergic rhinitis conjunctivitis, higher rates of allergen sensitization by skin prick test. Since then, many studies have been conducted that investigated this relationship between bronchiolitis in infancy and asthma development. And recently there's been a very useful systematic review and meta-analysis that shows that following RSV bronchiolitis compared to kids who did not have an RSV bronchiolitis, the risk for recurrent wheeze in infancy and at preschool age, but also the risk for asthma later on, increases by about a factor of 7. If these kids had a rhinovirus bronchiolitis compared to kids with an RSV bronchiolitis, that risk increases even further by a risk factor of almost 3. So the question that's been discussed for many years is whether viral bronchiolitis is just an early marker of asthma development that's happening anyhow, or whether it's a causal factor and contributes to the development of asthma. I'd like to show you 2 studies that make it likely that respiratory viral infections are a causal contributing factor. Excuse me, please. The studies are both from Tina Hartet's lab, and this one investigated whether in a large cohort that was followed for 5 years using routine clinical data, the authors asked whether the age of the children were at the peak of the winter virus season had an influence on the diagnosis of asthma at the age of 5 years. And it did indeed find that children who were between 2 and 5 months old at that time had a significantly higher risk to have an asthma diagnosis 5 years later than children who were younger or older during the winter viral peak. So suggesting that there is a period of susceptibility in that age. The other study was a prospective birth cohort where children during the first year of life were assessed every 2 weeks by ECR if they were RSV positive, and at the age of 1 they all had RSV serology done, which would be positive, completely independent of disease severity if they've been infected. And again, the question was if they had an asthma diagnosis at the age of 5 years. And interestingly, of the children who were RSV positive during the first year, 21% had an asthma diagnosis 5 years later, and that was significantly lower in the children who did not have an RSV infection in the first year. Again, showing that the first year of life is a period of susceptibility to an induction of asthma development if these children are hit, in this case by RSV. This group calculated that if it were possible to delay the RSV infection beyond the first year, there would be a 15% decrease in asthma diagnosis 5 years later. So their concept is that previously, let me just get a pointer up, that previously people primarily linked severe RSV infection with bronchiolitis to asthma development, and it may well be that this is both driven by genetics of the children, who makes severe disease to RSV and asthma more likely. However, if you include children with mild RSV or even silent RSV infection, just based on serology, that really makes it very likely that the presence of this infection actually has a causal influence on asthma development. So to really test this association, intervention studies are needed. And there's been one very important intervention study from the Netherlands, conducted in premature infants between 33 and 35 weeks of gestation, who were treated with the anti-RSV monoclonal antibody palivizumab during their first RSV season. And they were then followed up for a year with a median follow-up of 10 months. And the main outcome is, you can see this on the graph here on the left, are the cumulative days of wheezing during the trial. And you see that compared to placebo up here, the kids with asthma, with RSV prophylaxis had less than half the number of days with wheezing. Now, importantly, is this reduction in wheezing days was also evident outside the RSV season, at a time when the monoclonal antibody was not active anymore. And throughout the whole study, there was a reduction in recurrent wheeze by about 50%, from 20% to about 11%. The same group followed these children up further, up to the age of 6, and at that age asked if they had asthma. They assessed lung function, they assessed exhaled nitric oxide, IgE sensitization, and doctor-diagnosed asthma. And looking at these readouts, they came to the conclusion that RSV prophylaxis had no major effect on asthma development and lung function at the age of 6 years. However, they didn't highlight this, but they also reported a parent-reported outcome of current asthma, which was basically based on whether these children had had wheeze in the last 12 months, or whether they had been treated with asthma medication in the last 12 months. And here there was a significant difference. In the control group, 24% of the children did have current asthma by this definition, and the RSV prevention group only 14% had current asthma. So this is a relative risk reduction of almost 10%. And this is also apparent in the children who did actually have wheezing in their first year of life. Here as well, there is about a 13% relative risk reduction, making it, in my view, likely that this RSV prophylaxis, although it was in preterm kids, had some influence on asthma development. Now, more intervention studies are needed, and there are two scenarios where, well, natural is the wrong word, but I still use it, natural experiments will be done or have happened. One is that RSV prophylaxis with a long-lasting antibody or with maternal vaccination is about to be rolled out to whole populations of neonates, which will delay and reduce early RSV infections. And the other natural experiment were the lockdowns during the COVID pandemic. Just as an example here, this graph shows data from Western Australia, where the usual increase in RSV infections in young children in their winter, which you can see in the black line here, did not happen during the lockdown. And RSV infection only started happening when the lockdowns were released. This meant that the age at which children experienced their first RSV infection was delayed on average by 6 to 8 months. And it will be very interesting. So this happened not just in Western Australia, obviously, but there's data from many places that is very similar. And it'll be very interesting to see if this has an impact on the development of asthma subsequently. Right, so a question that I've been very interested in is which virus-induced responses in the airway mucosa can plausibly contribute to asthma development. And I could obviously speak about innate immune cells in the lung, about adaptive immunity, but today I really want to concentrate on the epithelial cells of the lung. The reason being, they are the primary target of RSV infection, and they are the first cells to develop an antiviral response. So, initially, here's some data from mouse models. In the mouse model of RSV infection, the epithelial barrier becomes reduced after infection. In studies we've done together with Chesniakis' group at CIAF, we've seen that tight junctions are downregulated, that protein influx into the airway lumen is upregulated, and this compromised barrier will allow the influx of allergens into the subepithelial layer where they can interact with immune cells, and it is likely to promote inflammation in the epithelium. Study by Saravia et al. In a neonatal mouse model, but then also in human infants, showed that the RSV infection leads to an upregulation of interleukin-33. Interleukin-33 is one of the cytokines that people also call alarmins, that are released by epithelial cells upon damage of these cells. And it is a cytokine that induces strong type 2 immune responses through the induction of type 2 innate lymphoid cells. And they also found that there is an upregulation of IL-13, one of the type 2 cytokines, which presumably came from these innate lymphoid cells. So So one question my own lab, or we've been interested in and looked at in my own lab, is whether healthy lung epithelial cells have immune regulatory effects, and if these immune regulatory effects may be lost after RSV infection. So we initially studied this in a mouse model. We took dendritic cells, antigen-presenting cells from mice, pulsed them with ovalbumin as an allergen, and co-cultured them with T cells that specifically respond to ovalbumin. These T cells were stained with a dye, and we were able to see that they proliferate because the intensity of the dye was diluted, and they also secreted cytokines. And then we've performed the exact same assay in the presence of lung epithelial cells, and found that these lung epithelial cells strongly inhibit T cell proliferation and cytokine production. And if we then repeated the experiment and infected these lung epithelial cells with RSV, this ability to inhibit T cell activation, T cell proliferation was lost. We then examined the same question in human primary bronchoepithelial cells, together with Irene Hein's group in Groningen in the Netherlands. So T cells were activated with antibodies in this case in the presence or without the presence of these bronchoepithelial cells. And again, the T cells had been stained, and here you can see on the left-hand side normal T cell proliferation. So each of these peaks is a division where the intensity of the staining gets less. And if the T cells don't proliferate, then you don't get this shift to the left. And you can calculate a stimulation or division index from that. So T cells by themselves stimulated with anti-CD3, anti-CD28 antibodies proliferated very well. But if they underwent the same stimulation in the presence of these bronchoepithelial cells, proliferation was largely abrogated. And then we treated the epithelial cells with a viral mimic, poly-IC, and then co-cultured with the T cells. And we found that the inhibition of T cell proliferation was lost. We also had the opportunity to look at primary bronchial epithelial cells, not only from controls, but also from asthma patients, and found that in the asthma patients, the ability to inhibit the T cell proliferation was less than in the healthy controls. So these data, I think, show that lung epithelial cells are potent inhibitors of T cell activation. I think they may be really important in maintaining a quiescent tolerogenic immune environment in the healthy lung. Upon infection with RSV or exposure to a viral mimic, these lung epithelial cells lose their ability to inhibit T cells. And interestingly, lung epithelial cells from people with asthma also have a reduced inhibitory capacity. So I think that this loss of immune regulation following a viral infection will, in all likelihood, facilitate T cell activation, not just in vitro, but potentially also in the lung. And that would, in all likelihood, also include the activation of allergen-reactive Th2 cells. And this may well contribute to excessive inflammation and immune responses, including type 2 inflammation in the lung following a viral infection. We were also asking the question whether there are longer-lasting changes in epithelial cells after RSV infection. And this is very recent data that Piotr Janas, a PhD student in the lab, worked on. So this is again in a mouse model. We took mice and infected them with RSV or with controls, and 4 weeks later we harvested the lungs, harvested the lung epithelial cells and analyzed them by PCR and then also by flow cytometry. And one of the really interesting findings is that the genes involved in the MHC class 1 and MHC class 2 complexes were significantly upregulated, not only acutely after infection, which I don't show you here, but also 4 weeks later. And Piotr then went on and looked at the protein expression of MHC class 1 and MHC class 2 4 weeks after infection using flow cytometry, and again found that after the RSV infection, both MHC1 and MHC2 were upregulated significantly, interestingly both in the epithelial cells from the airways and in the epithelial cells from the alveoli. So it seems likely, and this is something we're testing just now, that this upregulation of the MHC2 complex facilitates T-cell activation and T-cell responses, and this may well include responses to allergen again, and could again promote allergic inflammation in the airways. But just to summarize what I've told you about the effect of viruses on the epithelial cells and the mucosa in the lung, these red blocks are meant to be epithelial cells. If they are infected by viruses, you get several things. You get opening of the junctions between the epithelial cells, and allergens can get into the airways and interact with antigen-presenting cells much more easily. You get the production of alarmins, in particular IL-33, which activates these innate lymphoid cells, which in turn are producers of large amounts of the type 2 cytokines, interleukin 5 and 13, which lead to the attraction and activation of eosinophils, to mucus hyperplasia, and instruct the dendritic cells to interact with T cells in a way that furthers a type 2 immune response. And there may also be direct interaction between epithelial cells and the MHC2 complexes with memory or already committed type 2 immune cells, which will increase their activity and their function. Now, being an allergist, I can't help also briefly talking about allergens in the development of childhood asthma. So what's quite clear is that in children and young people, aeroallergens like pollen, house dust mite, pet dander are major contributors to asthma symptoms and asthma attacks. But what's less clear is whether they actually contribute to asthma development. And it's great that Steve Durham has already spoken on allergen immunotherapy, because allergen immunotherapy is a tool that may answer this question and may actually help reduce asthma development. So it's quite clear that in children and young people, allergen immunotherapy, be it subcutaneous or sublingual, can reduce asthma symptoms and the need for medication in children who already have existing allergic asthma. But interestingly, allergen immunotherapy has also been used in children who already have rhinoconjunctivitis, for example, to pollen, with the question whether this can reduce asthma development. And indeed, there are several studies that show that there's a potential to reduce the risk for asthma development in people who already are sensitized and have an allergic rhinoconjunctivitis. Now, the interesting question, which is still open, but there is initial data that I'll show you in a minute, is whether you can use allergen immunotherapy for primary prevention of the development of sensitizations and asthma. And a fairly recent study from the team of Hassan Arshad in Southampton was able to show that if children were treated with house dust mite sublingual immunotherapy, starting from the age between 5 to 9 months and treated over 3 years. The total sum of allergic sensitizations in these children was significantly reduced, and the diagnosis of asthma, according to a very rigorous algorithm at the age of 6, had a trend to being reduced significantly from 14% to about 3% of kids with an asthma diagnosis at that age. So, let me summarize what I tried to show you. Early life respiratory viral infections, importantly including mild ones, likely contribute to the development of childhood asthma. Virus-induced changes in respiratory epithelial cells can promote type 2 immune responses and inflammation. They may facilitate allergen sensitization. They can certainly be longer lasting, and I believe that they are likely to contribute to asthma development. And I just showed you that allergens contribute to asthma. I think allergens contribute to asthma development in children and young people, and allergen immunotherapy can prevent, as secondary prevention, asthma development in people who already have allergic rhinic conjunctivitis, and it may be able to prevent primary allergen sensitization and asthma development at a very young age. Let me briefly show you some people in the lab. I showed you data primarily from Piotr Janas and all the data from Hongwei Wang. So thank you very much to my lab, to collaborators and our funders. And thank you for listening. I'll leave you with 2 pictures from Edinburgh, where I am at Edinburgh University. Thank you.
Thank you, Dr. Schwarze, for a wonderful